In the textbooks, they talk about the nucleus controlling the cell. And that's in today's textbooks, in grammar school through medical school. And the nucleus has the DNA, and the DNA is believed to be the control. As I said before, every function in your body is already present in every cell. So where you have an organ in your body, the cell has an organelle, which means miniature organ. So the textbooks talk about the nucleus is equal to the brain. Watch. If you remove the brain from an organism, the organism dies. But if you remove a nucleus from the cell lives and the behavior is unaffected. Some cells can live 2 or more months with no genes, and they carry out all of their normal functions. So enucleation is not removing the brain. So the nucleus is not the brain. But the nucleus does something. When you take out a nucleus, the cell cannot reproduce its parts or itself. So the nucleus is not the brain. The nucleus is the gonad. Since science is a male business, and since men think with this organ, they made it the brain of the cell. The nucleus does not control the cell. The nucleus is reproduction of the parts. But we went and looked for to find all of the genes that make up a human. And the Human Genome Project was going to be the last biology project. Because once we knew all of the genes, then we would be able to control anything in a human being. It takes one gene to make a protein. There are 150,000 different proteins. How many genes do you need? 150,000. How many genes did we find in the Human Genome Project? 23,000. There's a problem. There are not enough genes to make a human being. So what is wrong is our belief. We are completely wrong about our belief that genes control life. So genes do not control life. The fact. So assumption number 2, false.
Wrong.
Wrong. Good.
I'm learning.
In my research, 40 years ago, I started working with stem cells. People think that stem cells are something new. But I learned very many things from the stem cells. The secret of life I learned from stem cells. Right now, in every one of you, you have stem cells throughout your entire body. And these stem cells can replace any tissue or organ in your body. And you should be able to stay young for hundreds of years, as Greg will talk about. So you might ask, then why do we age? And the answer is because the mind controls the cells and the genes. And we collectively believe in aging. So that my research, early research, I have a tissue culture with multipotential stem cells. These are clones, meaning they're all genetically identical. In my experiment, I take a group of cells, so I take this group of cells and put it into another petri dish with a different environment, environment A. And environment A, the conditions of the medium, the stem cells form muscle. But I could go back and take the very same genetically identical cells and put them into culture dish with environment B, and the cells form bone. Or I could go and take the very same cells and put them in culture dish C with a different environment, and the cells form fat. What controls the fate of the cell? The environment. They were genetically identical, only the environment was different. So I published this work in 1977. And it's a fine structural analysis of normal and modulated cells in myogenic culture. It sounds like Latin, but it sounds like Latin. And so science is like the church, so you can't understand what I'm talking about. But what is the meaning? Is that this is the first scientific report that I made, how the environment controls the fate of your cells. And I use the word modulated, meaning altered, because there was no other word. My colleagues were not interested in this work because they were interested in the human genome. So I left I had tenure, and I walked out of the university as a professor. I left the university because they were not being scientific, because they didn't want to understand this. So I end up at a better university, the Stanford University Medical School. There I repeat the experiments, but more sophisticated. And I get the cover of the journal, Differentiation, but I really wanted the cover of Rolling Stone, but it's Science. So after I do this, again, the scientists were not interested. So I left the university for the second time. And I write my book on the biology of how cells work. And in my chapter 2, I lay out the scientific evidence to show you genes do not control biology. I also introduce you to the exciting discoveries of epigenetics, a new field of biology that is unraveling the mysteries of how the environment influences the behavior of cells without changing the genetic code. And I call chapter 2, It's the Environment Stupid. It's a takeoff from President Bill Clinton's campaign saying it's the economy stupid. And 5 months after I write this, people are saying, what do I know? I'm not at a university, so what do I know? And then Nature, in one of the most prestigious journals in the world, Nature, 5 months after my book, an article about stem cells are engaged in a constant crosstalk with their environment. Biologists are fast realizing. No, some of them not too fast. What do you think they call this article in Nature? It's the same story. Science is now saying what I've been saying for 30 years is coming around now, and that there's a new science of how cells work. But this is going to be the fun part, because this is where I'm going to explain how the biology actually works. And I will we'll get to the secret of life before we're over. Okay, so what we were talking about is how cells actually work. And it's very similar to this. It's a machine. So just as the gears move and connect and turn and make an action, so do the proteins inside the cell. And there are proteins, I said there are 150,000 different proteins that provide both for our structure and our behavior. So 150,000 different shaped proteins. Now, again, it's like inside the cell, they're just like a clock with all the proteins engaging like gears. So this picture is going to show 3 different examples of proteins. These are molecules blown up very large. And the question is, what makes each protein a different shape? So where does the shape come from? And is underneath the skin, there's a backbone inside each protein, a backbone. And just like a backbone gives you your shape, or you can change your shape, so does a protein. Now, what's really interesting is that all proteins share the same characteristics, so I only have to talk about one protein, but then I'm talking about all proteins. All proteins have a backbone made out of linked units, just like the vertebrae. The backbone pieces are called amino acids. There are 20 different shaped amino acids. And what makes each protein different is the length of the chain and also the sequence of the different amino acids. The DNA, the gene, is the information of which amino acid comes after the other amino acid. But this is not the best model because all the beads have the same shape. So I'm going to use 3 different shaped pipes. A straight pipe, one with a 90-degree bend, and one with a 45-degree bend. That's just 3 out of how many? So, okay, and they're just like the beads, they plug into each other. And these are bonds, and in the real chemical name, they're called peptide bonds. So I will assemble this one protein. And now you can see that the sequence gives a rigid backbone. And one quick question. If I take this apart and put it back together again in a different sequence, will I get the same shape? No. So good, because that means what's the point is that every protein has a unique sequence. So how does it work? Let's go to this picture. Can you in your mind see them moving? Okay, now replace the gears with protein. Okay. Now in your mind, can you imagine when this protein moves, it moves this protein, and then it moves this protein. And this is a combination of a protein and a metal machine. Now take away the metal parts, protein machine. Now, there are different groups of proteins. When they move, they make different functions. And the proteins are called pathways. Okay? So there's respiratory pathway, a digestion pathway, muscle contraction pathway. Okay, so inside every cell are protein gears. When they move, they make functions. And most students in medicine or science or complementary medicine study this protein system called the Krebs cycles. Anybody knows the Krebs cycle? And these are protein gears that when they engage create the function of the Krebs cycle. Okay, now are you ready for the secret of life? Okay, before I tell you the secret, I want you to know the sky is not going to open up and the light come in here. It's not such a big secret. Like a backbone, you can twist and flex and change the shape. So I will show you 2 shapes and ask you which shape is more stable, 1 or 2. And you can't answer the question because I leave out one fact. Okay, the yellow pipe are both negatively charged. 1, or this is 2. 1, which one? This one. Yeah, because the 2 negative charges, they repel each other. Okay, now the secret. There's a protein, and then there's something called a signal. The signal can be a chemical, like a drug, a hormone, a growth factor. Or a signal can be a vibrational wave. But I'll use the chemical one because you can see it. Now here's this is negative, the yellow, okay? And this is positive charge, but more positive charge than this has negative charge. Okay? This comes like this. And when it gets near the protein, it'll bind. What's the charge at this end? Yeah, and the charge here? Is this more stable or is this one more stable? What did it do? It changed shape? It moves. Movement is where life comes from. When proteins change shape, they create behavior. Now, if the signal comes off, what does the protein do? It moves back. So that you are made out of proteins, that gives you the structure, but the movement is the protein plus the signal. So if you are dead and you still have your protein body, what's missing? The signal. Okay, so in this protein gears, and each protein is like a lock, and the signal is like a key. And there's a different signal for different proteins. When the signal moves and binds to the protein, behavior. When the signal is not there, no behavior. If you want the behavior to go again, what do you have to do? Another signal. Another signal, and then protein goes again. Your body will move when the signals come, will stop when the signals are not there. So these are, again, the three different proteins, and these are actual molecular studies of how the protein moves. With signal 1, with signal 2, signal 3. And this is the behavior of protein. Now, in medicine, the signals are always indicated to be physical chemical, because medicine is Newtonian. So all signals must be matter. Recently, physicists are looking at protein movement. And this is a paper in the journal Nature. And these two authors, Popfristic and Goodman, were studying how proteins move, the behavior. And they wanted to predict the movement. So like Newton, when he predicted the movement of the planet, they used Newtonian physics, a Newtonian equation, to predict the movement. It didn't work. And this is a review of this paper. And this chemist, physical chemist, writes, the most pressing question raised by Popfristic and Goodman, when will chemistry textbooks begin to serve as aids rather than barriers to this enriched quantum mechanical perspective on how molecular turnstiles move? Because when they use quantum physics to study the movement, they were able to accurately predict the movement. So the most important conclusion is the subtitle of the article. What are the forces that control the twisting and folding of molecules into complex shapes? Don't look for the answers in your organic chemistry textbook. The reason why this is important is that medicine is based on organic chemistry, and the answers to how life works are not in the book. In a more recent one, this concept of materialism, matter only, Bosch. Okay. This is a more recent paper just a few months ago, and how they control biological functions. They were studying protein movement, and what they showed was this. It acted as a quantum mechanical machine. And if they shine a light on the protein, when the waves were in phase, the protein was active. And when they shine a light and the waves are out of phase, it shut off the protein. So it says, for a quantum mechanical object, one can arrange interference of several paths to create constructive interference that selects one state and destructive interference that blocks the other. Proteins respond to good vibes and bad vibes. So this is a picture of the light pulse that they shine.